What's up? Okay, we're going to get started. We know that several people will be coming late, and I know there's a large number of people that'll be attending remotely. As many of you know, this is a special grand rounds for us, both because of the speaker that we have today, and also because it is really a memorial grand rounds in honor of Ray Adams, who, as this audience and everyone watching remotely knows, was the preeminent neurologist of the 20th century and the chair of neurology at the Mass General Hospital for about 27 years before stepping down in 1977 and Joe Martin taking his place. In that time and in the several decades after, before his death, he was a pioneer of just about every branch of neurology and made seminal discoveries in just about every area of adult and pediatric neurology, and is also the author of the preeminent textbook, Principles of Neurology, along with Maurice Victor, which is in multiple editions even since his death. So again, we're really pleased to be able to honor him today, and also to introduce our speaker today. We're very privileged to have Dr. Randy Schekman. Dr. Schekman did his PhD with Arthur Kornberg at Stanford, followed by a postdoc with Seymour Singer at UCSD, and has essentially spent his entire career in the state of California, where the weather is much better than it is in Boston these last few days. For the most part, after he finished his postdoc, he spent, I think, his entire career of almost 50 years at UC Berkeley, and he's, as you all know, has done seminal work in terms of understanding the very basics of membrane trafficking within cells, initially starting with yeast, but of course moving on to many other systems since then. And as you can imagine, Dr. Schekman has received numerous awards for his outstanding achievements over the decades, including most notably the Albert Lasker Award for Basic Biomedical Research in 2002 and the Nobel Prize for Physiology or Medicine in 2013. Dr. Schekman is also known as a pioneer in medical publishing and biomedical publishing, and was not only the editor-in-chief of the Proceedings of the National Academy of Sciences for a while, but also was the founding editor of eLife, and again, a pioneer and staunch advocate of open access publishing since that time. Also, in more recent years, he's moved his studies into Parkinson's disease, which he'll talk about today in terms of his research talk, but he also has a very prominent position now in leadership, and that he is the scientific director of the ASAP program, which is Advancing Science Across Parkinson's. And that's done in conjunction with the Sergey Brin Foundation, as well as with the Michael J. Fox Foundation, and has provided, I believe, hundreds of millions of dollars in research support for innovative studies in Parkinson's disease. So it's a real pleasure for me to welcome Randy here. I think it's his first time at the Mass General Hospital, and it's a real privilege for us to be able to have him visit us today and to listen to his talk. So Randy? And we do have a plaque in honor of this special occasion to present to you. You want to take a photo? Okay.
Thank you very much. I'm delighted to be here today, to be speaking to a dedicated personal audience and presumably to a lot of people out there in the ether. This indeed is my first time at MGH, but my wife, who died of Parkinson's disease, was a nursing student here over 50 years ago in the Bullfinch. So I have an emotional connection to you. Well, I am not a neurologist. I'm not even a physician, but I do biomedical science, and my biomedical science career has touched on subjects of some considerable interest in normal and disease human physiology. I'll talk about two topics relevant to this, and which are both being pursued clinically, and that is, the first part of my talk will be about extracellular vesicles, how they're made, and what their function may be. And then I'll talk about part of the work that I'm doing as part of this program, ASAP, that has to do with the unconventional secretion of a protein that's certainly crucial in the progression of Parkinson's disease, and that's a protein called α-Synuclein. So let's begin. First, by tradition, I gather that it's important to stipulate my conflicts of interest. And so here they are. I have my research support comes from the Howard Hughes Medical Institute, from the Michael J. Fox Foundation, and from the Sergey Brin Family Foundation. I also serve on a variety of biotech advisory boards and prize boards, none of which have any financial involvement in my own research, which is purely basic science. So let's begin then. For those who may not be aware, there are two broad pathways for the production of extracellular vesicles made by all cells. Even bacteria make extracellular vesicles. But I'll focus our work on human cells grown in culture or in human physiology. There are vesicles that bud from the plasma membrane. They are often called microvesicles. They arise by processes that are used by envelope viruses such as HIV to pinch from the cell surface, capturing, in the case of a virion, a ribonucleoprotein particle, or in the case of microvesicles, simply sampling cytoplasm from the cell, including cytosolic proteins and RNA. Then there's a specialized pathway that produces vesicles through a branch of the endocytic pathway, where cell surface receptors are internalized for downregulation, often for destruction in the lysosome. But at the same time, cells take up cytoplasm, and in certain cases, very selectively, microRNAs are captured into vesicles that pinch into the interior of a multivesicular body. We've been studying several such microRNAs that are highly enriched, though with low chemical concentration in exosomes, and have studied the role of several different RNA binding proteins. I'll refer to one in a moment called LA. The lupus LA antigen is an RNA binding protein that has a role in the selective capture of certain microRNAs. These microRNAs are then secreted if the MVB happens to fuse at the plasma membrane of a target cell. I'll talk about the forces that reinforce that targeting event, and that results in the secretion of a bolus of exosomes, which are characterized by vesicles that have a diameter of approximately 100 to 150 nanometers. They have certain highly enriched membrane proteins, tetraspanins, but also other proteins that are diagnostic of the origin of these vesicles. And they include cytoplasmic proteins, some enriched, and a variety of small RNAs, tRNAs, and a very small population of highly selected microRNAs. Now, we've devised procedures to resolve the two broad populations of extracellular vesicles using a linear optoprep buoyant density gradient. A former graduate student in my lab, Moraima Timosh Diaz, was able to show that a low buoyant density population is marked by certain proteins, some of which are unique to low buoyant density vesicles. I'll have more to say about what gets packaged into these vesicles in a moment. And then a higher buoyant density population of vesicles that are typically highly enriched in a tetraspanin CD63. Now, we find that although in conditioned medium and certainly in blood plasma samples, there are other particles that are often confused with exosomes. Many of them are simply ribonucleoprotein particles that separate from vesicles on a buoyant density gradient. They stay at the bottom. And so there have been claims in the literature, we believe they're incorrect, that microRNA processing enzymes such as dicer and argonaute were claimed to be inside of vesicles and to continue processing in that step. But we find that these large particles can be separated quite nicely on a buoyant density gradient. Now, let me summarize a paper that's just now about to be published, the work of Jordan No, a terrific graduate student in my lab, who's carried on from the studies that Timoshe Diaz did on the sorting of RNAs, microRNAs, mediated by the LA RNA binding protein. I'll show you some data, but let me summarize what Jordan found. He was able to show that although we know that LA interacts directly with several microRNAs through a 3' uridylate, tract of uridylate residues, we now have found through proximity labeling that LA, after interacting with the RNA, interacts with what had been assumed to be a receptor P62 for autophagy, and that in turn interacts with a lipidated protein called LC3, another autophagy protein. And this sandwich of 3 proteins with bound RNA is internalized into exosomes to be secreted from cells. Now, some of the data for what I've just described is shown here. The detection of P62 associated directly with LA was shown by using an apex fusion to LA incorporated into vesicles, where we could see that LA was also incorporated. When the P62 gene, this is its genetic designation, is deleted from cells, it no longer is detected, at least in a crude supernatant fraction, although the LA protein is still present and abundant. But when vesicles are isolated by immune precipitation from the LA deletion cell, from the P62 deletion cell, LA is largely removed compared to the control. So there's a significant depletion. Now, what's particularly interesting in the context of what I'll talk about next is that in exploring the variety of microRNAs that are affected by deletion of P62, we found a set, quite a larger set than this, that are not affected by deletion of P62, and another set, some of which become highly enriched in cells that are defective in P62 and largely depleted from exosomes secreted by those same cells. Now, in this case, the cell of interest that we've done this with is MB231, a breast cancer cell line. And we noted among those microRNAs that are depleted from exosomes secreted by P62 null cells, a preponderance of microRNAs that in the literature have been associated with tumor suppression. And so this seems to suggest that the cells have relied on a mechanism for their selective release from the tumor cell so as, we think, not to interfere with normal tumor progression by this cell line. This conforms to a view that we've had about extracellular vesicles, which I will discuss as the story goes on, that they are not, as often is believed, produced to deliver content to target cells, but rather their main function may be in cellular homeostasis. Their main function may be to get rid of things that the cell no longer wants or doesn't need, or may interfere, in the case of a tumor cell, may interfere with its normal tumor progression. Thus, the quite selective and striking secretion of tumor suppressor microRNAs that is blocked in P62 null cells. Now, for the next few minutes, I'm going to focus on our studies on the origin of extracellular vesicles. And for this, I need to take a step way back to over 100 years ago with an observation, a more recent example of which is shown here, but an observation that was made by developmental biologists at the turn of the 20th century, that it's possible to rupture the plasma membrane of an egg, in this case a sea urchin egg, with a needle, which initially is accompanied by an explosive release of cytoplasmic content. However, 30 years ago, it was observed that if the incubation is continued in the presence of millimolar concentrations of extracellular calcium, the damage produced by the needle is quickly repaired so well that the egg cell can go on and be fertilized and turn into an embryo, and then in this case, into a mature animal. So there's a very active plasma membrane repair pathway. This pathway has been studied for some years by many investigators, but principally by Norma Andrews, who was for many years a faculty member at Yale, but moved her team to the University of Maryland. She and others showed that when cells are damaged by any condition that allows calcium, extracellular calcium, to enter the cell, she showed that the lysosome is the first organelle that is mobilized through an interaction with calcium binding proteins and delivered to the site of lesion to initiate a patch repair pathway. There are many ways in which she and others explored how this works, which I don't have time to review, but in appreciating her discoveries, two of my students, one, the first that I've already mentioned, Jordan, and another student, Justin, came to my office now a few years ago with what was a really intriguing idea, and that is we know that the lysosome membrane is very similar in composition to the membrane that surrounds a multivesicular body. And so what Justin and Jordan suggested was that the same conditions of damage that would elicit a mobilization of the lysosome may do so for the recruitment of the multivesicular body to the plasma membrane. And if this were so, then conditions that cause calcium to enter the cell at millimolar concentrations would result in the rapid secretion, at least of exosomes, as a consequence, as a byproduct of a plasma membrane repair pathway, not necessarily for any function that the vesicles would themselves serve, but rather as a byproduct of a repair pathway. All right, so now how to test this? Well, our typical analytic procedures to evaluate the composition and formation of exosomes involved very large scale, at least for us, large scale collection of plates and hundreds, sometimes a liter of conditioned medium. But that seemed really not very practical for the experiments that Justin and Jordan wanted to do. So they came up with a clever and simple assay that allows us to detect in just small microliter samples of conditioned medium vesicles that are sealed and which have an enzyme, luciferase, expressed facing the interior surface of the exosome. The way they did this was to take advantage of the CD63 gene, to which at its C-terminus, a copy of N-luciferase was fused by a Japanese group. The N and C termini of the CD63 protein are predicted to face the lumen of an extracellular vesicle. That is, they would face the cytoplasm of an endosome, and on invagination into the interior of the endosome, they would then face the lumen of an extracellular vesicle. But as anyone knows who works with cell culture, there's a lot of debris and fragments of membranes that are shed into the growth medium. And so they wanted to find a way to eliminate the signal that might be attributable to fragments of membranes or even N-luciferase that happened to be secreted unconventionally. And so what they did was they found a membrane impermeable inhibitor of the luciferase and a membrane permeable substrate, a colorimetric substrate. And the combination of these two allows the detection fairly uniquely of only the luciferase that's sealed within the vesicle. So we're only measuring the sealed content, and that eliminates a high background. So the first experiment, which gave a striking result, was on treating these cells with ionomycin, a calcium ionophore, for as few as 10 minutes. Elicits a rapid burst, a 30-fold increase in the secretion of, in this case, of exosomes. And this is dependent on extracellular calcium. Other even more vigorous treatments, such as treating cells with sublytic concentrations of a bacterial toxin, streptolysin O, at concentrations that are still sublytic in the presence of calcium, but not in its absence. These permeabilized cells continue to secrete exosomes, in this case, a 20-fold increase in detected exosomes, Other experiments where they simply pipetted cells through a narrow gauge needle that introduces just excessive stress on the cell surface also elicit a striking increase in the secretion of exosomes. So this was really quite interesting. So we decided to have a look at what calcium binding proteins might be invoked to mobilize, in this case, multivesicular bodies in these cells that were treated with ionomycin. We used procedures that Norma Andrews and her colleagues had already used, where we were able, in this case, to immobilize endosomes and lysosomes by an epitope tag on the membranes of both in the presence of beads, but with no antibody. No lysosomal membranes are detected in the bead pellet, but on addition of calcium and the antibody on the beads, they were able to show a set of calcium-dependent proteins, a number of which had already been identified associated with lysosomal mobilization, and a family of proteins called the annexins, well-known calcium binding proteins that have been implicated in plasma membrane repair through the mobilization of lysosomes, also appear to be involved in MVB formation, because when we used shRNA to ablate the expression of one of the annexins and one of the proteins that was found, copine-3, another calcium binding protein, in both cases, eliminating the expression of these two proteins greatly reduced the ionomycin elicited secretion of exosomes. So this makes the case that these proteins not only involved in mobilizing the lysosome, but also almost certainly involved in mobilizing exosomes in this case. Now, we then re-examined the distribution of extracellular marker proteins on the buoyant density gradient that I showed you in my first data slide. And in cells that were treated with streptolysin O, the vesicles that were secreted sediment quite clearly into two classes. First, the high buoyant density vesicles characterized by CD63, which we ascribe to exosomes, but also quite a substantial increase in the low buoyant density vesicles, the so-called microvesicles, in this case characterized by 3 different annexin proteins. Much more of these vesicles are secreted, as well as of exosomes, by treatment either with ionomycin or with streptolysin O. So this was quite striking. And Justin decided to do a thorough quantitative mass spec analysis by fractionating vesicles after treatment with, in this case, ionomycin. And 2 populations based on their buoyant density separation were evaluated by mass spec. And indeed, we found among the low buoyant density vesicles, quite a number, probably all of the calcium binding proteins that had been discovered to have a role in mobilizing the lysosome are found associated with these low buoyant density vesicles. And all the ones that we detected were inside of the vesicles. They were not on the outside. They were inside the vesicles, as though they had been recruited to the plasma membrane, and then through some pinching reaction at the plasma membrane, they remained enclosed inside of the vesicles. In contrast, CD63 and other proteins known to participate in exosome biogenesis or in the formation of a multivesicular body are found in the high buoyant density vesicle fraction. Now, in one particularly dramatic experiment, it was possible to visualize one of these annexins being recruited during a brief laser rupture of the plasma membrane, recruited quite specifically enriched to the region of damage and included in the vesicles that are shed from the cell surface under these conditions of physical damage. And so if you follow the arrow, you'll see about a micron laser ablation of the cell surface, followed by a clear enrichment of a scarlet labeled form of annexin 2, not only to the plasma membrane, but shed in membranes released from the cell surface. And this is followed within just a couple of minutes by a complete repair of the damage and the cell goes on its merry way. Now, we were able then to quantify the release of both populations of vesicles using, as I've described previously, the N-luciferase fusion to CD63, shown here over time, and treatment with sublytic concentrations of streptolysin O, and an even more dramatic release of annexin 2, marked by a fusion to N-luciferase, also dependent on a chemical concentration range of streptolysin O. We've now gone on and have been able to study the sequence of events in the recruitment of annexin and of other proteins that leading to the culminating recruitment of escort 3 proteins that pinch the vesicle from the plasma membrane, just as Wes Sundquist has shown is required for the pinching of HIV particles from infected cell surfaces. So we think that there's a natural pathway that is not dependent on virus infection that is used to recruit these proteins through introduction of millimolar concentrations of calcium damaging the plasma membrane. Early this year, we published a review article in the Angiobiochemistry where we've come to the conclusion that this process that I've just described may be quite be responsible for a major source of extracellular vesicles of both types, both of exosomes and of microvesicles. There are many conditions in the body, which you people will know much better than I, that induce some forms of shear on cells, the shear that we could reproduce simply by pipetting cells through a narrow gauge needle happens in the body all over when cells migrate through cell boundaries where through shear forces that are brought about by flow through capillaries, blood clots, platelets are known when they participate in a blood clot reaction to secrete quite abundant exosomes and microvesicles. And also muscle damage. It's known, for instance, that after vigorous physical exercise, the level of CD63, an exosomal membrane protein, rises in the blood of people who've done controlled experiments. So there are really lots of reasons that we enumerate in this review article to suggest that extracellular vesicles, far from having a particular role in the body, may largely be produced as an important byproduct of repair mechanisms. And so we look forward to a vigorous discussion and dispute about this model in the years ahead. All right, in the remainder of my talk, I'd like to turn to my efforts in 2 directions. One, as a role in leadership of a program organizing groups around the world to study basic aspects of the cellular, molecular, and systems roles in the initiation and progression of Parkinson's disease. And then in the effort in my own lab to understand how one of the proteins that's genetically and certainly clinically implicated in Parkinson's disease, α-Synuclein, how it may be unconventionally secreted from cells and how that may be the basis of the spread of α-Synuclein in the brain of patients who progress with the disease. So the program that I become involved in is called Aligning Science Across Parkinson's. Craig mentioned at the outset what this is. It came about, in my case, because my wife died of Parkinson's now 8 years ago. And when she died, I was approached by the chief financial officer of the Sergey Brin Family Foundation to ask if I would have an interest in helping them develop a program of basic science. Mr. Brin is the co-founder of Google, enormously wealthy, but regrettably, he has a family genetic history of Parkinson's. He has a dominant allele in the gene LRRK2, one of the 20 or so genes known in familial forms of the disease. He shares this with his mother, who is quite ill, advanced illness with Parkinson's. He is asymptomatic thus far, but he is at some significant risk. And so he has, over many years, invested hundreds of millions of dollars in the Michael J. Fox Foundation, which I'm sure you're all aware of, a wonderful organization that supports patient groups and clinical research. They have a small budget for basic science, but Brin quite independently decided that what we really needed with Parkinson's after 200 years since it was first diagnosed, there's really nothing that's been developed to stem the inexorable progression of the disease. And he decided that what we needed was to understand it at a very, very basic level, basic science. So I was asked to lead this effort, which I've been thrilled to have the opportunity to do. I'm going to focus on α-Synuclein, which was already, even before this, the subject of some of the work in my lab. And I'll now review some aspects of the disease that I'm sure you're all familiar with. So sorry if I bore you for a few minutes. But as you know, the midbrain has a region, the substantia nigra, that can be detected in sections because of the production of melanin, an oxidative product of catabolism of dopamine. This unique deposit is detected in this case, a section from a patient who died for other reasons. With patients who die of Parkinson's, most often this region is no longer detected because it's likely that by the time they die, most of the dopaminergic neurons have died, and thus no melanin is being produced, at least not at this location. 100 years ago, again, I'm sorry to bore you with this, you neurologists will know this, a German physician, clinician, Friedrich Lowy, discovered in sections of the nigra cells, dopaminergic neurons, that were highly enriched in a cluster of aggregated material, which we now call Lewy bodies. The Lewy bodies form likely through the aggregation of α-Synuclein. Alpha-synuclein is indicated here in a simple linear diagram. It's a small, relatively unstructured protein, 3 alleles of which are known among familial forms of the disease. These potentiate aggregation of α-Synuclein. But even just a triplication of the gene in the rarer form of the disease, triplication of the normal alpha-synuclein gene is enough to cause alpha-synuclein to accumulate and to begin what is a likely buildup of other things that are seen in Lewy bodies, not just alpha-synuclein, but other protein aggregates, including membranes and filaments that produce structures that occupy a considerable fraction of the cytoplasm. Now, it still remains, as far as I'm concerned, controversial about whether Lewy bodies are the basis of pathology. They certainly correlate with pathology, but it's only just now, maybe this year, that they're developing PET probes that will allow us to visualize these structures in patients as opposed to in biopsies. So there is a correlation. Whether it's a dead-end product that cells use to preserve themselves and some other event related to the buildup of these that causes the cell death is not clear, but nonetheless, they are still a hallmark of the disease. Some years ago, a German clinician by the name of Brock, again, I'm sure many of you are aware of his studies, found in sections of patients who died at different stages in the disease, a seeming progression of Lewy bodies from the brainstem, possibly originating in the vagus. There are many people who believe of a gut origin for Lewy bodies, but I have to say the evidence that I've seen for this is not very persuasive at all. There are animal models that show apparent progression of Lewy bodies from the gut to the brain, but those experiments, frankly, are not easily reproduced in different laboratories. So again, I'm getting a little skeptical about the gut to brain axis for progression of the disease. Nonetheless, Lewy bodies in patients who die early in the progression of the disease, Lewy bodies are sometimes detected in the olfactory bulb, and this may explain, as was the case for my wife, that sometimes decades before the symptoms, the classic symptoms emerge, people experience a loss of their acute sense of smell. And a surprising fraction of patients who have this, certainly not a majority, but a surprising fraction, report when they are diagnosed with Parkinson's to have a poorly developed sense of smell. This now is becoming a standard test that's being applied to patients who may be interested in and may have some family history of Parkinson's. So there's an organization that we support, PPMI, Parkinson Progression Monitoring Index, that has collected now thousands of patients, and they send out scratch and sniff tests for people who may be experiencing some symptoms that they are concerned about. And so we're now being able to follow these patients over a very long term. So it may be, and this is just a correlation, that the Lewy bodies may explain this loss of acute sense of smell. As the disease progresses, the Lewy bodies can be seen in other sections elsewhere in the brain, eventually up to the cortical regions where emotional and cognitive disturbances, a very large fraction, again, I don't need to tell you, a very large fraction of Parkinson patients experience dementia. This was certainly true in my wife's case. And it may be because of the accumulation of Lewy bodies in the cortical regions of the brain. So the idea that we had, and many others have had, is that the spread of alpha-synuclein, giving rise to these Lewy bodies elsewhere in the brain, may be the result of some secretory process. But alpha-synuclein is not a traditional secreted protein. It has no signal peptide. There's nothing about it that looks like it would pass through the normal secretory pathway. And yet, it can be detected in the blood. It can be detected in CSF. It's clearly getting out of cells. So the question that we've asked is, how does it get out of cells? There are other small proteins that cells secrete unconventionally, and there have been various pathways developed to understand how this happens. At the very core of it is the question of which membrane the protein passes as a prelude to its secretion from cells. How does it get across the membrane? Is there a channel, a different channel than is used for the normal secretory pathway through which proteins like alpha-synuclein may pass that would allow it to be secreted out of the cell? And could this be an explanation for the spread of alpha-synuclein as Parkinson's progresses? And if so, could there be some therapeutic intervention that would block this unconventional path and ensure that the accumulation of alpha-synuclein could be dealt with in other ways, such as by degradation through the proteasome in the cytoplasm or through degradation in the lysosome? So we've done a number of experiments, first with HEC293 cells, and then with more traditional dopaminergic neurons to investigate the mechanism of secretion. And for those of you who note this, when I make presentations at the Howard Hughes Medical Institute, we're not allowed to name the journal that we published the papers in. Many of my Hughes colleagues put the PMID number here, which I think is kind of silly because no one will write that down. But I've decided to take a different approach to emphasize that I don't publish in Cell, Nature, or Science for reasons that I can explain later. So the simplest possibility for secretion unconventionally would be it somehow gets to the plasma membrane and is secreted soluble into the extracellular medium, or that it could be secreted in an exosome, and perhaps these exosomes can pass from cell to cell. So there's a simple way to test that, and that is to use differential centrifugation. I'm going to tell you about the work of Shenji Wu, postdoc in the lab, under conditions where we detect alpha-synuclein in the growth medium. It is not sedimentable. Only a very small fraction of it is sedimentable. It's almost entirely in the supernatant after a length and speed of sedimentation that would pellet all extracellular vesicles. So we've then examined the size of, and this is under normal conditions, not with a pathologic mutant form of alpha-synuclein. Wild-type alpha-synuclein is secreted purely as a monomer from these cells. Now, for proteins that pass across the membrane, it's generally found, but not always found, that they have to pass in a relatively unfolded form through a channel. In the ER, it's a channel called Sec61. In the mitochondrion, there are channels in both the outer membrane and the inner membrane. And in both cases, proteins have to remain conformationally flexible. to pass across these bilayer as they're being made or threaded in a linear fashion through the channel. And so we asked that simple question for α-Synuclein and got a very different answer. In this case, what we did was we made gene fusions between α-Synuclein and a 3-helix bundle protein constructed by the David Baker lab in the University of Washington. A protein was designed to be virtually resistant to denaturation. You can boil the protein at 100 degrees for 10 minutes and it doesn't unfold. If you fuse that protein, the triple helical bundle, to a mitochondrial precursor protein, the presence of that unfolded domain blocks the mitochondrial protein from entering across the mitochondrial envelope. But in contrast, what we found is that if you fuse this unfoldable bundle to either the N or the C terminus of α-Synuclein, it continues to be secreted into the growth medium, as you see in this panel in the middle, in abundance similar to when it's secreted without such an impediment. And so this suggests that α-Synuclein, in passing across a membrane, somehow no longer requires an unfolded state. There are a few examples of proteins like that in the cell. For instance, proteins that go into and out of the nucleus have to pass through the nuclear pore, and that happens when the proteins remain folded. Very recently, Tom Rapport here at Harvard has shown that proteins that enter into the peroxisome similarly can pass through the membrane in an unfolded state. And the way in both cases this happens is that the channels through which these proteins pass have a special interior that's formed by a kind of hydrogel-like substance that allows large structures to pass as long as they are provisioned with a signal that allows them to gain access to this hydrogel and to pass through it. That may be what's responsible for α-Synuclein, but we don't know. And so what we've decided to do was to try to focus in on finding the membrane channel molecules or candidates for molecules. And to do so, we first turned to the literature and found that there is a chaperone protein known first in neurologic disease, and secondly, to be involved in the secretion of α-Synuclein, a chaperone protein called DNA JC5. This we've studied and published. It's a small protein that is lipidated, it is palmitoylated, and we found it associates with the outer surface of the endosomal membrane. And we've shown, as is indicated in the data that I'll now describe, that DNA JC5 really is actually required for the unconventional secretion of α-Synuclein. And it does so apparently through mobilizing α-Synuclein into the endosome. And when the endosome then is delivered to the plasma membrane, α-Synuclein can be secreted by that fusion event. The way we demonstrated this was to rely on techniques, a slightly different technique than the one that I've described using ionomycin. We found that if you treat cells with baflomycin 1, a drug that blocks the ATPase in the lysosomal and endosomal membrane and causes these organelles to become neutral in pH, that itself seems to mobilize the secretion of exosomes, as shown in the panel on the left, by a quite substantial level, just as I showed you earlier when cells are treated with ionomycin. So we use this to investigate the pool of α-Synuclein that might be mobilized, in this case by treating cells with baflomycin. And we found that we could induce the secretion of α-Synuclein, in this case about 5-fold, by treating cells with baflomycin. And when cells had the single DNA JC5 locus deleted, there was no longer such stimulated secretion. So we conclude that DNA JC5 is somehow involved, possibly in recruiting α-Synuclein to the surface of the endosome, but it's not a membrane protein per se. It's lipidated, and so it can tether to the cytoplasmic surface of the endosome, but it couldn't possibly be a channel. It may be serving as a chaperone, but it's not a channel. So to frame the next experiments that I'll describe, we found that the DNA JC5 actually forms an oligomer, and we believe that alpha-synuclein somehow interacts with this oligomer and is positioned on the surface of the endosome to be exposed to some kind of integral membrane receptor or channel through which alpha-synuclein gains access to the endosome. So now I'll tell you about an experiment that Shinji cooked up that has highlighted a family of membrane proteins known in the literature for some years that we find enriched in the endosome that have apparently a direct role in this traffic event. The way he looked for such proteins was to use a variation on biotinylation, where you can take, in this case, something called turbo-ID, which is another one of the biotinylating enzymes. And it's possible to make fragments of this turbo-ID and to reconstitute biotinylation activity by fusing these fragments to two proteins that you believe have some direct interaction. And by bringing them together, the fragments of turbo-ID recombine and then can biotinylate proteins in proximity to the two bait molecules that are set up in this way. So this was the experiment, very simple one, where we express the two fragments, in one case fused to alpha-synuclein under conditions where I've shown you does not impede its secretion. And in another case, to DNA JC5, where we've shown it also does not impede its function in promoting alpha-synuclein secretion. And when they are together, we ask what membrane proteins become biotinylated by their presence. And this worked really well. And the major protein that's seen is actually DNA JC5 that gets biotinylated when both fragments of the turbo-ID are present, but not when just one or the other is present alone. So what else gets biotinylated? We harvested membranes, washed them, solubilized them, and isolated all the biotinylated membrane proteins and subjected them to mass spec. And the surprise was that in addition to DNA JC5, a family of proteins called the SCAMPs, secretory associated membrane proteins, were identified. These were first identified actually in synaptic vesicles in Tom Sudhoff's lab, but since have been studied in a number of contexts where they have some as yet ill-defined role in secretion. There are 5 paralogs in the human genome. There's one, SCAMP5, that is a brain-specific one. SCAMPs 1 and 3 are very abundant in HEC293 cells. These proteins are at micromolar concentrations in the cell, so that's enormously abundant. As you'll see in a moment, we can show that these, the 3 most abundant ones, can be detected in endosomes. We think these 3 are particularly easy to detect by biotinylation because these 3, 1, 2, and 3, have N-terminal cytosolically exposed domains with lysine residues that are targets for biotinylation, whereas SCAMPs 4 and 5, the brain-specific one in particular, has no such exposed domain that would be readily biotinylated. So what do we know about the SCAMPs? Well, their function really has been rather elusive. There have been many suggestions, but nothing really definitive. They're highly conserved, even in plants. They are regrettably not present in yeast, or else somebody would know something about their function, but they're in plants and animals quite ubiquitously. You'll see in a moment that Drosophila has a single SCAMP, as does C. elegans. SCAMPs 1 and 2 and 3 are major isoforms, and as I said, SCAMP5 is a brain-specific one. Using endosome lysosome immunoprecipitation with an epitope that has been tagged with 3 HA copies, you can see that SCAMPs 1, 2, and 3, for which we have unique polyclonal antibodies to detect, are highly enriched in the immunoprecipitated, but relatively less abundant in the input fraction, precipitated along with LAMP1, a major lysosomal membrane protein. And other membranes are not so precipitated by this procedure. In an AlphaFold model, the SCAMPs are tetraspanin membrane proteins. There's no obvious channel. If you look at just a single monomer, but we know from experiments I don't have time to describe that these proteins form quite stable oligomers, and it remains possible that the oligomeric form has a channel. As I mentioned, SCAMPs 1, 2, and 3 have a prominent N-terminal cytoplasmic domain and a shorter C-terminal cytoplasmic domain. Now, 2 experiments that we've been able to do suggest some function for these proteins. Just for the time being, all I said was that they're in close proximity to alpha-synuclein and DNAJC5, but that doesn't say anything about its function. So there are 2 experiments in human cells that we've done. We've used a neural progenitor cell that's differentiated from neuroblastoma cell line SY5Y, differentiated in the presence of retinoic acid to bruise neurons that are dopaminergic-like. We found that when SCAMPs 2, 3, 4, or 5 are overexpressed in differentiated neural progenitor cells, there's a measurable increase in the secretion, the unconventional secretion of alpha-synuclein, whereas there is no such effect on the release of cytoplasmic protein as a marker protein. So there's clearly something going on that makes the SCAMPs a bit rate limiting, surprising since they're so abundant. Conversely, when the SCAMPs are knocked down, we noted among the molecules of alpha-synuclein in the cytoplasm of these differentiated cells that there's a substantial increase in the detection of a phosphorylated form of alpha-synuclein. Serine 129 phospho-alpha-synuclein has been implicated in pathology. It's found in Lewy bodies, and it's found in the brains of patients who are, experimental animals that are models of Parkinson's. There's a somewhat of an increase in the insoluble P129 phosphorylated alpha-synuclein in the SCAMPs 3 and 5 knockdown, and you can see here that we are very efficiently knocking these proteins down. So it may be rate limiting, and when you knock out or down any one of the paralogs, there's clearly something going on that renders alpha-synuclein a substrate for aggregation. The P129 phosphorylated alpha-synuclein is often found in oligomers in cells where it's detected. But that doesn't really get to the real answer that we want to have, which is what are the SCAMPs actually doing? Is it a channel? Is it a receptor? What is the molecular nature of its role in this process? And for that, we've turned recently to a collaboration with Mel Feeney, who has a lab here in, I think, in Brigham's. She has for some years been used Drosophila as a model for Parkinson's disease. There are reports of mutations in genes. I think it may have a LRRK2 gene that shows some motility defect. She, in her previous work, showed that if you express human alpha-synuclein in the fly brain, the cells have a slower but not eliminated ability to crawl, as you'll see quite graphically in a couple of minutes. So there's some interference with some brain function and motility when you simply produce human alpha-synuclein in the fly. Now, she found out in the literature, when she heard about our work, that there is in the literature already, there are 2 deletions of the single SCAMP gene. They are broad deletions. They are apparently null in respect to the detection of an immunoblot form of the protein, but they may not completely eliminate at least other fragments of the protein. So they may be hypomorphs of some sort. The work that's published shows that they have some neurologic defect, but not any particular motility defect. And as you'll see, she also found no motility defect. So let me now share with you the results of quite a striking experiment that she's done at the postdoc in her lab, Vanitha Nithanyan has done. If you look at normal flies in a tube here, they move around quite nicely. If you look at the SCAMP mutant, one of the deletion mutants, their motility is essentially the same. That's true of both deletions. No effect, at least as measured by this kind of motility, visual motility assay. If you express α-Synuclein in the normal fly brain, there's a reduction in motility as quantified here. But if you do so in one of the SCAMP deletion mutants, the flies are paralyzed. Quite a striking defect in motility in these flies. So there's clearly something going on about SCAMP function in α-Synuclein. Now, if you visually inspect with an antibody against the human synuclein that's expressed in the brains of these flies as a control, without expression of the human synuclein, there's no detectable immunofluorescence. But with expression, likely a higher level than would normally be present, in a human brain, one sees cytoplasmic accumulation of α-Synuclein, and she's shown independently that this is a phosphorylated form of α-Synuclein that accumulates in the presence of when the protein is expressed in the brain of a normal fly. In the SCAMP mutant flies, there is clearly a quite abundant accumulation of α-Synuclein, largely apparently in the cytoplasm in both of these deletion mutants. Again, consistent with the idea that SCAMP's having some role in getting α-Synuclein into the endosome and then out of the cell, or somehow out of the cell. All right, now I'll leave you with a model. It's still very speculative, but at least it's something that we can sink our teeth into. We're pretty sure that α-Synuclein gets into the endosome somehow. And we know that DNA JC5 and perhaps the SCAMPs participate in this, but we can't say for sure how the SCAMPs do so. One possibility is that it's a channel. The SCAMPs form an oligomer that may present a channel through which α-Synuclein progresses. The channel may be sufficiently flexible to allow an unfolded domain to pass, in which case it would be soluble in the endosome, and on fusion at the plasma membrane, the α-Synuclein would be released, largely monomeric, along with extracellular vesicles, and may also be released by this condition. Now, alternatively, another model which I tend to favor is that the SCAMPs are serving as some kind of a receptor for α-Synuclein, doing so in the context of being bound to the DNA JC5 protein. We argue that the phosphorylated form of α-Synuclein may cause the synuclein to also bind to another receptor, P62, on the surface of the endosome and be internalized somehow through autophagy or some other process into the interior of the endosome and then be translocated, eventually secreted from the cell. So the question remains, how is it that α-Synuclein gets into the endosome? How does it cross the membrane? Even if the SCAMPs are receptors, there still is the challenge of getting the protein, even with some brick of helix bundle bound to it, across the bilayer. This remains a fundamental problem that we hope to explore in the coming years. All right, let me leave you with the group. This is a group at a department retreat some years ago. I started my work by introducing the work of Moraima Tamosh Diaz, former graduate student, now in the biotech industry. The team of Jordan No and Justin Williams, remarkable, two terrific graduate students. I'm so proud of what they've done. Really all by themselves have come up with the idea that I presented about damage to the plasma membrane being responsible for secretion of extracellular vesicles. And more recently, Shenji Wu, and now current and recently departed postdoc in the lab, who's done all of our work on α-Synuclein. Now, I take special pride in not only in the achievements of my group, but also of their sense of humor. And this was illustrated to me quite dramatically in a picture I'll show you in a moment in my parking garage, where I have a special parking place right next to another special parking place for Jennifer Doudna. We get the privilege of our own parking places. Not to be outdone, Justin decided to take a picture of my parking space and replicate it here for his own private use as a privileged graduate student, requiring no special permit whatsoever. He parked here for 2 weeks without being detected by the parking cops and didn't get a ticket at all. So, and I didn't even notice it myself. So I'm so proud of him. Okay, I think probably I'm done. So happy to take questions. Thank you.
Well, thanks, Randy. That was great. One question I had, and maybe I know there's been papers on this, but what does synuclein do normally?
Yeah.
And how is that impacted by this need to get rid of it, under these like cell damage states?
Yeah. I don't think it's entirely clear what it's known. It was discovered initially as a protein that isolates along with synaptic vesicles. It's on the surface of synaptic vesicles. It may be facilitating some aspect of synaptic vesicle presentation or fusion. Deletion of alpha-synuclein has only a very subtle effect. There are 2 paralogs, which can, in certain measurements, compensate for alpha. There's beta and gamma. But clearly, alpha-synuclein has this unique propensity to aggregate, not shared by beta and gamma. So its normal function is still not entirely clear.
Well, thanks for the brilliant talk. I don't think this would ever work here with the parking lot. Probably the first 10 minutes they will figure it out. So in terms of strategy to translate on disease-modifying therapies for Parkinson's disease, is it possible to target the unconventional alpha-synuclein transport and release and so on versus strategies for clearance?
Yeah.
Right.
Yeah. I mean, when I got funding from the Michael J. Fox Foundation, I presented this as a different opportunity for some possible therapeutic application. But again, my lab is entirely basic science. We'll proceed to try to figure out if SCAMPs are a channel or if not something else, and we'll leave it to other people to see how that might be applied. I am most excited about clearance as a means of getting rid of alpha-synuclein more efficiently. And so in the latest round of funding that we've now just recently decided on, there are a number of groups who have different approaches to the clearance mechanism. One group that's unfortunately not represented in our teams is your colleague at Harvard Medical School, Bruce Spiegelman, who has a really interesting story about clearance. I don't know if you're aware of his work, but Bruce, over a period of years, has discovered an exercise-induced peptide hormone called irisin. It's a proteolytic cleavage of a normal cell surface receptor molecule that is expressed in the brain, or at least brain penetrant. And he has shown that, for example, if you take cortical neurons, primary culture into which you've introduced additional copies of alpha-synuclein so as to accumulate aggregated forms, that administering alpha-synuclein, administering irisin results in the complete clearance, likely by autophagy of the aggregated alpha-synuclein. So he's started a company and he's got his lab now devoted to trying to see if this natural human peptide hormone could be a treatment for that would promote clearance. I'm very excited about that effort, and I will follow it, even though it's not going to be part of the ASAP program.
Thank you. Thank you. Very interesting talk. So my question is about the first part of your talk with the calcium signaling and the idea that exosomes might be excreting things that are unwanted. I'm curious, I'm a neuroimmunologist, so the immunology piece has been, people have talked about MHC1 and 2 being in exosomes. Do you think that you could think of a mechanism, we know that MHC presentation is partly calcium dependent, that you could propagate the immune response that way, or similarly TCR activation, causes intracellular calcium entry. Do you think that's a relevant mechanism?
Yeah, so the idea would be that MHC would then spread in exosomes to other cells. Well, that has some appeal, but we in other experiments, and I didn't really emphasize this, have shown that although exosomes can be abundantly internalized by target cells, they end up just being degraded in the lysosome. There's really very little evidence that EVs of any sort are efficient delivery vehicles. So any mechanism that would require MHC being implanted in the plasma membrane of a target cell would require some kind of membrane fusion event. And the problem with EVs is they lack a fusogen, a known fusogen that would be responsible. Unlike enveloviruses, EVs don't have an obvious membrane fusogen. So there's an awful lot of hype in this field about EVs being delivery vehicles. They may be signaling vehicles. They may be decoys. They could serve many. They could carry small molecules and spread them as they diffuse. But I don't think they are efficient delivery vehicles.
Thank you.
Thank you for the great talk. I have to say sorry, I was wrong about α-Synuclein seeding positivity. By saying positivity, I actually had a negativity in my mind.
Yeah.
Sorry about that. And a question for the second part of the talk. Do you think the secreted soluble soluble can see the aggregation in healthy cells?
Yeah. Not likely by itself. And we discussed this earlier. There's something, maybe since you're probably mostly neurologists here, you must know, just within the past couple of years, we finally have a biomarker for Parkinson's, and that is the presence in CSF of Parkinson patients, mostly the sporadic onset forms of the disease, of a factor, some likely protein factor, that stimulates the aggregation of alpha-synuclein. The correlation between the presence of this seeding factor and the disease is really quite pronounced. And it's detected even in prodromal patients with RBD. It's less abundant in some of the genetic forms like LRRK2 for reasons that aren't fully understood, but in the sporadic forms, it's there. And so we'd love to know what that seeding factor is. It's probably not soluble monomeric. One team who's progressing this by trying to purify the factor has shown that if you take CSF that has seeding activity and you centrifuge at 100,000 G for an hour, it still remains in the supernate, and so it's not a vesicle, it's not big. But if you put it on a gel filtration column, the activity filters as though it's a complex of about 1.5 to 2 million Daltons. Whether it's a small oligomer of alpha-synuclein plus something else remains to be seen. This is something that the program will pursue. So I think it's a critical question.
Time for one more? Okay. I want to return to this idea of what's the normal function of alpha-synuclein that's remained mysterious despite so much work on this protein. And you may also be aware of Vic Khurana, who runs a lab at Brigham and Women's, has a really interesting paper suggesting it may have a role in mRNA stability and expression. But the mechanism that you're starting to uncover here for how alpha-synuclein monomers are taken up into vesicles seems pretty involved. And I guess my question to you is, do you think that's a general mechanism by which a bunch of proteins are uptaken into vesicles? Or do you think that's specifically designed to bring alpha-synuclein into vesicles? Yeah. And does that suggest that there may actually be a role for alpha-synuclein excretion that's been designed to excrete it in, or do you think that's just a byproduct of?
I think that I'm pretty sure there'll be other clients for this mechanism. One is tau. So we've sent our cell lines that are deficient in DNA JC5, and another group at Stanford has found that tau secretion is greatly reduced in the DNA JC5 mutants. They haven't looked at SCAMP, but I'm betting SCAMPs will be involved also in tau, which would be very interesting. So this could be a really powerful target, that a broader application than just with PD. Of course, that's remains to be seen. But I'm guessing if one were to do a survey of secreted proteins in the presence and absence of DNA JC5, you'd see a set of those that depend on DNA JC5 and likely on SCAMPs. So that's certainly worth doing.
Any final questions? Oh, one more. Okay.
Hi. I had sort of two related questions. One being, would you reckon that this excretory pathway is something that is cell type specific, or, I mean, I might just not be
It's quite general. Even yeast cells have an unconventional path of secretion that involves some other proteins that people have found to be required for unconventional. There are different pathways of unconventional secretion. We've looked at cytokines, IL-1 beta, and found a different membrane protein called TMAD10 is required for the unconventional secretion. We found that in that case, IL-1 beta first enters into the intermediate compartment between the ER and Golgi, and then progresses into an autophagosome and is secreted in that way. So it's a very different kind of pathway, we think, than what we found for alpha-synuclein. And the former postdocs who are now back, we've done this work back in Tsinghua and Beijing, have found that TMAD10 deletion reduces the unconventional secretion of a whole bunch of other proteins, including of other cytokines, in cells that are not producing this protein gasdermin, which is normally responsible for the release of cytokines in non-immune cells. There's an alternative pathway that seems to involve TMAD10 and possibly as a channel.
Thanks again, Randy. I think that was our last question. So we really appreciate you coming. Thanks.
Okay.